Hello learners, till now we have learned about the chemistry, metabolism, digestion and the sources of lycopene. Today we will be learning about metabolism, bioavailability and effect of processing on lycopene. As we all have studied in our earlier part, lycopene belongs to a group of carotenoids and found mostly in tomatoes, red grapefruit, red guava, to name few. Most plentiful carotenoids in human plasma include beta-carotene, alpha-carotene, beta-cryptoxanthine, lutein, zeaxanthin and lycopene. These 6 major carotenoids account for approximately 70% of all carotenoids identified in human plasma and tissues. Let's now understand the metabolism and bioavailability of lycopene. The objective of the present module is to understand the metabolism of lycopene, how the lycopene will be bioavailable and effect of processing on lycopene. Let's now first see the metabolism of lycopene. Lycopene were found to be cleaved by beta-carotene 9,10 oxygenase BCO2. Interestingly, the recombinant ferret BCO2 catalyzes eccentric cleavage of cis-lycopene isomer effectively, but not all trans-lycopene at 9 and 10 double bond. In addition, cis-lycopene may act as better substrate than all trans beta-carotene for ferret BCO2. The mechanism whereby the ferret BCO2 preferentially cleaves the 5-cis and 13-cis isomer of lycopene into apo10 lycopenal, but not all the trans lycopenal is currently unknown. One possible explanation is that the chemical structure of cis isomer of lycopene could be mimic the ring structure of beta carotene molecule and fit into the substrate enzyme binding pocket. Although this hypothesis warrants further investigation. The observation that supplementation of all trans lycopene result in significant increase in cis lycopene tissues concentration in ferret underlies the significance of this observation. Previously, few scientists have identified a group of lycopene, a oxidative product 2,6-cyclolycopene 1,5-diol A and B in human serum and many other tissues including prostate, lungs and colons. Recently, a series of apolycopenals including apo10 lycopenal have been identified in human plasma. Hope learners, you have understood the metabolism of lycopene. Let us now see the bioavailability of lycopene. Lycopene bioavailability is dependent on number of elemental factors like food processing and dietary composition. Lycopene can occur in different form in fresh plant food including carotenoid, protein complex in chloroplast or in crystalline form inside chromoplast. The effect of processing and storage on lycopene structure and stability are of interest for number of reasons. Improper processing and storage like exposure to light and oxygen may affect the lycopene isomers which may completely degrade, making these food products less desirable for the customers and consumers. It has been observed and concluded by many studies that thermal processing generally improves lycopene bioavailability by disturbing cellular membrane, which allows lycopene to be released from tissue matrix. Now, lycopene is a lipid soluble compound. Consuming it with fat increases its bioavailability. For the example, consuming salad with full fat dressing results in higher blood carotenoid level than eating salad with reduced fat dressing. In vivo studies demonstrated that cis isomer of lycopene appears to be more bioavailable than all trans isomer. In vitro experiment support the conclusion that increased bioavailability of lycopene cis isomer is at least partially due to increased Isomerization and increased uptake by enterocytes relative to all trans lycopene. Human organ stores lycopene to different degrees. Lycopene found in highest concentration in liver, testes, adrenal gland and adipose tissues. It is found in lower concentration in kidney, ovary, lung and prostate. The amount of lycopene content increases as tomato matures. The lycopene concentration is higher in summer and lower in winter. Now let us now discuss the effect of processing on lycopene. With an increasing understanding on health benefits of lycopene, much of the work is undertaken to preserve the loss of lycopene during processing and also to understand the maximum availability of lycopene with which process. Lycopene belongs to carotenoid family and heat, light, oxygen, diverse food matrix have effect on lycopene, isomerization and auto-oxidation. Lycopene may isomerize to mono or poly cis form with the presence of heat or oil due to hydration. It has been seen that temperature higher than 100 degrees centigrade and longer heating time leads to larger percentage of lycopene degradation. Few research studies support that presence of oil during food preparation may increase the percentage of mono or poly cis lycopene in the food. All trans lycopene was found to decrease with increased illumination time. Approximately after 144 hours exposure to light, it resulted in 94% loss of lycopene. The lycopene loss was highest in presence of air and light at 25 degrees centigrade and lowest under vacuum and dark. Now dehydrated tomato products are sensitive to color fading and loss of acceptability mainly due to lycopene isomerization and oxidation. When lycopene emulsions were diluted in the food, lycopene was found to be very stable. Hope learners, you have understood the metabolism, bioavailability and effect of processing on lycopene. Let's now conclude with the past few years we have gained a greater knowledge of metabolism of lycopene, biological activity of lycopene derivatives. In particular, characterization and study of beta-carotene 9,10 oxygenase has shown that this enzyme can catalyze eccentric cleavage of lycopene to form apo-10 lycopenoids. This raised an important question whether the effect of lycopene on various cellular functions and signaling pathways is a result of direct action of intact lycopene or its derivative. Several reports support the notion that biological activity of lycopene can be mediated by apo-10 lycopenoids. More research is clearly needed to identify characterized additional lycopene metabolites and their biological activities, which will potentially provide invaluable insight into mechanism underlining the effects of lycopene in human. We have studied the improper processing and storage. Many affect the ratio of lycopene isomer and degrade lycopene entirely, whereas thermal processing generally improves lycopene bioavailability. Lycopene is a lipid-soluble compound. Consuming it with fat increases its bioavailability.